Hello everyone, I am Swastika and welcome to my presentation. The topic of my presentation is resistance to fluoroquinolones. The fluoroquinolones resistance in bacteria primarily arise through target site mutation and efflux mechanism. Mutation in the quinolone resistance determining regions QRDRs of DNA gyrase GYRA and GYRB and topoisomerase 4, that is PARC, PARE, alter the drug binding site, preventing fluoroquinolones from attaching and blocking DNA replication. Alongside these, efflux plums such as NORA in Staphylococcus aureus and OQXAB or QEPA in gram-negative bacteria actively transport the antibiotic out of the bacterial cell, lowering its intracellular concentration and thereby reducing its effectiveness. Another important resistance pathway is plasmid-mediated quinolone resistance, which enables horizontal gene transfer between bacteria. Plasmid often carry QNR genes encoding proteins that seal DNA gyrase and topoisomerase from fluoroquinolone binding. Additionally, plasmid-bound efflux pumps such as QEPA and OQXAB further enhance resistance by expelling the drug. Together, these mechanisms create a multi-layered defense system that not only reduces fluoroquinolin's efficacy but also facilitates the spread of resistance across different bacterial species. Fluoroquinolones are a class of broad-spectrum antibiotics that are widely prescribed in both human and veterinary medicines because of their ability to treat a variety of infections. They are especially effective against urinary tract, respiratory, gastrointestinal, and systemic infections. However, the increasing emergence of resistance has led to treatment failures, limiting their effectiveness. This poses a significant challenge for healthcare systems since fluoroquinolones were once considered reliable first-line drugs. Resistance is now common in several clinically important bacteria including E. coli, Salmonella, Pseudomonas, and Staphylococcus. This problem extends beyond hospitals as resistant bacteria can spread through animals on the food chain and humans, making it one health concern that links human, animal, and environmental health. Due to the rapid global rise of fluoroquinolone resistance and its public health implications, the WHO has identified it as a major global threat that requires urgent monitoring and control strategies. Resistance develops from chromosomal mutations in GYRA and PARC after repeated fluoroquinolone use. Plasmid-mediated genes spread resistance between bacteria. Overuse of fluoroquinolones in agriculture and animal husbandry has accelerated this process. The mutation selection window drug level between MIC and MPS allows resistant mutant to emerge. Phenotypic tests include disk diffusion and MIC determination for nalidixic acid, ciprofloxacin, and levofloxacin. Molecular method to determine it is by using PCR to detect GYRA, PARC mutations, and PMQR genes, or sequencing for detailed analysis. Clinical cutoff guide interpretation shows that MIC-CIP should be greater than 0.12 microgram per microliter, suggest for resistance. For nalidixic acid, MIC-NaL should be uh, greater than or equal to 32 microgram per ml, indicate resistance. Newer fluoroquinolones, moxifloxacin, gatifloxacin cover a broader spectrum but resistance can still occur. C8 methoxy FQS plus B-lactamase are useful against Pseudomonas to limit resistance. Combination therapy with beta-lactamase or efflux pump inhibitor improve efficacy. Keeping drug labels above MPC helps prevent emergence of resistant strains. Resistant genes QNRA, QNRS, OQXAB, QEPA are often carried on ESBL plasmids, causing multi-drug resistance. PMQR frequently coexist with beta-lactamase genes like BLACTX, BLATEM, and BLASSB. Such gene combinations make bacteria highly resistant and harder to treat. Animal to human transmission has been reported from poultry, cattle, and pig. Thank you.